Bismillahirrahmanirrahim. Uh, this is the method to analyze the types of blood cells in rabbit. First of all, we have apparatus for the practical, that is, we need slides, distilled water, 90% alcohol, and Lessman's stain dropper, petri dish, and compound microscope, and a rabbit. Next is the procedure. First of all, we took the glass slides and then wash the slides with 90% of alcohol, and then placed blood drops of the rabbit on the slide, and then produced a smear of the blood, and then allowed the smear to dry for a minute, then placed the slides in a petri dish, and put some drops of Lessman's stain on slide and covered the petri dish, and then uncovered the dish and threw twice the volume of distilled water to the stain and keep it for 10 minutes, and then we drain out the slides and wash the slides with distilled water, and then slides are observed under compound microscope. This was the procedure for the practical, but before this there is a specific method for the blood collection in a rabbit. We do not collect the blood blindly, but there are specific places from where we collect the blood from the rabbit. The two primary vessels used for blood collection from a rabbit are the marginal ear vein, and yielding to small to moderate quantities of blood depending on the experience and expertise uh, of the performer, and the central or uh, auricular artery from which a larger volume of blood can be collected, and other veins or sites that may be used include the lateral uh, saphenous vein of the hind leg, and the cephalic vein on the foreleg, and the jugular vein. There is a method for collection of blood, that fill an examination glove with water and tie it off, microwave it until its temperature is warm. मीन हम इसे गर्म करेंगे इतनी देर तक कि इतना गर्म करेंगे कि इसे टच किए जा सके, एंड देन अप्लाई दैट टू रैबिट्स एरा फॉर अबाउट अ मिनट. फिर इसे रैबिट के अप्लाई करेंगे, एंड देन scrape the skin over the purposed vein puncture site with oil of the wintergreen, and then collect blood 1 to 2 minutes after its application, taking care to rinse any residual oil of the ear after the blood has been collected. ब्लड कलेक्ट करेंगे ईयर से, एंड देन administer SC promazine subsequently, about 15 to 20 minutes before blood collection. The time involved is perhaps not practical in a busy practice. Gently stroke milk the vessel with the thumb and forefinger from the base of the ear toward the tip of the ear. Recommended needle size range from 22 to 25 gauge, and selecting the smallest gauge needle required to minimize discomfort and tissue trauma. Then we observe following types of blood cells and following composition of blood in the rabbit. Um that are blood cells are red blood cells, white blood cells, and platelets. And white blood cells may be granulocyte, agranulocyte. Granulocytes are neutrophil, eosinophil, and basophils, and then agranulocytes are lymphocyte and monocytes. Then next we have analysis for the uh, amount and concentration of these blood cells in the blood sample that can be studied in this slide. You can observe it by yourself. Thank you for watching video. Allah Hafiz.